Right, guys. Let's talk membranes. Before we do that, we're just going to quickly look at their building block, the main kind of component of the membrane, the thing that's holding everything together, the phospholipid. Right. So, quick recap of your biological molecules. The lipid. What is it? It's made of 3 molecules. And there's 2 types of those molecules. So, first one is glycerol. Okay, so it's a 3-carbon molecule, and each carbon has an OH group on it. Okay, so that's glycerol. And for simplicity's sake, though you might not agree with me, what we'll do is just put one of those OHs up there, and you'll see why later. Okay, right now, a triglyceride has 3 fatty acid chains connected to the glycerol, but a phospholipid has 2. Now, what am I talking about? Well, each of these OHs in a phospholipid is connected to a long hydrocarbon chain via an ester bond. So that's glycerol, and it's connected to 2 fatty acid chains. Now, when we talk about fatty acids, what we're talking about is something like this. And this zigzag line just means that there's a carbon here, a carbon there, carbon there, carbon there, carbon there, carbon there, carbon there. It's a long chain of carbons, and each of those carbons is not hydrated, but hydrogenated. Okay, so it's got hydrogens on it. So it's a long hydrocarbon chain, and it's very non-polar. It doesn't interact with water. This whole part is very hydrophobic, and this is really important. So this part is hydrophobic. This part is called the carboxylic acid group. Okay, and so I'm going to re-simplify this again. So just know in your mind that after this carboxylic acid group, there's just a long chain of carbons. Each carbon has got hydrogens attached to it by covalent bonds. And the charge, the electron distribution in this part of the molecule is very evenly distributed. So there's no regions of positive or negative charge, which means it's hydrophobic. Another way to describe it might be non-polar. And because it's non-polar, it's actually hydrophobic because it won't interact with water. Okay, that's very important. Now, what happens is that there's going to be a let's see how this works. There's going to be a condensation reaction here, and that will lead to the bond, a new covalent bond forming between the fatty acid and the glycerol, and the loss of water. So that water will come off there, and we'll have a new covalent bond between the oxygen and the carbon. Right there. And that bond is called an ester bond. Okay? So, this, now we've lost the carboxylic acid, so it's no longer carboxylic acid. And this is called an ester bond. Right? Okay, so that's an ester bond. So the glycerol is linked to a fatty acid via a condensation reaction to form an ester bond with the loss of water, and the reverse will also happen. So we can add water and hydrolyze it, and this usually happens during digestion when we are consuming fats, and they are broken down in our digestive system to be absorbed into the blood. Okay, so this happens twice. Okay, so with the second OH group, we have again another what do we have on this side? Another fatty acid connected, and so we have two hydrophobic kind of tails, and we have this head up here. Now on the third OH group we get a phosphate connected. Okay, so a phosphate group attaches itself on the third one, again via an ester bond. Alright, so we get the oxygen, and this is the phosphate group right here. I've drawn it right. Yes, I think I have. Okay, and that will have a negative charge on it right there. Okay, so let's just look at the main components that we have. I'll draw this bit in red because this is really important. So this is a phosphate group right there. Okay, that's the phosphate group. And as you can see, it's got a negative charge. It's got oxygens and a hydrogen there. So there's going to be lots of uneven charge. That hydrogen, remember, is partially positively charged because it's connected to the very electronegative oxygen. Oxygen with negative charge there. So all this is charged, and because it's charged, it makes it highly hydrophilic, and therefore it will interact with water. Okay. In the opposite case, down here, the fatty acid tails, the hydrophobic tails of the lipid are non-polar, very evenly distributed electrons, evenly distributed charge, therefore hydrophobic, and does not interact with water. Alright, so you've got one molecule here, the phospholipid, hydrophilic, hydrophobic, one molecule with, kind of two very different personalities, as it were. And this is what's important. So if we're going to draw this in a kind of very simplified way, that's essentially what we have, okay? And what we need to remember is that this is our head, and remember that this is hydrophilic, and these are our long hydrocarbon tails, and they are very hydrophobic. Really? Yep, okay, hydrophobic, yeah. Okay, so that's what we have to remember, that this one molecule has got has got parts with very different properties, and that's essentially what leads to the formation of the membrane, as we'll discuss next, okay? Right, just a side note, you won't often find a phospholipid as simple as this, because usually this oxygen here has got something else attached to it, which we'll just call X for now, because it could take a variety of different forms. So you'll usually find, if you look up the structure of a phospholipid, you'll usually find something else attached to that oxygen. So it doesn't mean that it's not a phospholipid, but phospholipid just has this kind of general structure. Just like amino acids have R group that can change, not change, but there are different types of that R group. In the same way, you can get phospholipids, and that oxygen usually has something else attached to it. Don't need to know that, all right? Just remember, hydrophilic phosphate group attached to the glycerol. Glycerol also attached to two fatty acids, which give the lipid its very hydrophobic properties. All right, okay, so we'll come back and discuss next how this leads to the formation of the membrane structure. Right guys, so let's take a trip back in time, many millions of years ago, even before life as we know it evolved. Back then, we're thinking that, very simple basic molecules or sorry, not basic, very simple molecules such as lipids might have been floating around at that time. Okay, now they must have been around. Now, the thing is that if you have many lipids floating around, this situation that they are floating around in the water is not ideal for their stability, because these hydrophobic tails are having to interact with water, as we can see here. Not so much of a problem for the hydrophilic heads, because they are polar, they are hydrophilic, they are stable in their interaction with water. So, what's more stable than this situation is when these lipids associate in a very particular way. Namely, if they all associate in this way, I hope you can see that now we have a more stable situation. Okay, we've got all the hydrophilic heads interacting with the water, and we can show that they are more stable by drawing a smiley face there. Okay, and the tails are also more stable because they're pointing their hydrophobic tails away from the water by forming this little structure right here, and so those hydrophobic tails don't have to interact with the water. And the hydrophilic heads can interact with water. Yay! On its own, however, this is not big enough to form a cell. So as you know, if this structure got bigger and bigger, the unavoidable situation is that we do have water on both the inside and the outside of this layer. So that's going to be a problem. So if you give me a few minutes, hopefully this won't take too long. In fact, I'm not going to waste your time. Okay, so we are back. We talked about the very small kind of stable structure that lipids could form to make to keep their interactions with water optimized in that way. But as our structure would get bigger, we would encounter a problem, because not only would it have to deal with water being on the outside of the membrane, but it would also have to deal with water being on the inside. You can't avoid it. You can't have a vacuum in there. You have to have something in there. And in a watery environment, such as most organisms are, blood plasma, tissue fluid, cytoplasm, all these things, they're essentially water-based. So you can't escape water. Most of your cells are surrounded by water and have water in them. So how do you form a barrier that's going to satisfy both the lipids both on the inside and the outside of that environment? And the way is this. So you have this. Now these fatty acid tails are not going to be stable. That interaction is not going to be stable, because obviously these tails are hydrophobic. The heads on the outside interacting with water, no problem. What about the inside? The inside is also watery, so another layer of lipids can arrange themselves on the inside, facing their or pointing their hydrophilic heads to the watery environment on the inside. Okay? And what this does is, that means that the hydrophobic tails can interact with each other in this structure, because that environment there is hydrophobic. So both of these layers of lipid are satisfied by their respective interactions. So the hydrophobic tails of this layer are interacting with the hydrophobic tails of the other layer. The hydrophilic heads on both layers are interacting with the watery environment. Okay, so I hope you can see how the formation of what is now called a bilayer bilayer of lipids satisfies watery environments outside and a watery environment inside, which is needed because if this very primitive thing was to develop into a cell, it would need to have proteins in there, it would need to have amino acid, it would need to have nucleic acid, and those things are all water soluble. So if this primitive thing has any chance of becoming a living organism, it has to have a watery environment inside. And the only way that that can be satisfied is by forming the bilayer of lipids. Okay, so I hope that kind of makes sense. All right, so we are calling this structure a lipid bilayer. Yep, and what we are going to do now is have a look at what it looks like in 3 dimensions. Okay, so this is a kind of cross section. Right, what we're going to do is then just have another look at what this might look like. Yeah. All right, so I'll join you in a few minutes. Okay, so this is another way to look at what we've just talked about. If we took a small section of that kind of sphere that we were looking at and looked in detail, this is what we'd see. We have one layer of lipids here at the top facing the outside, and we have the watery environment on the inside. I'll call it in, but remember what we're referring to is the cytoplasm here. Okay, or the cytosol, whatever. Okay, so we have one layer of lipids there with the hydrophobic tails pointing inwards and the hydrophilic heads facing the watery environment there. And again, we have the other layer with the hydrophilic heads pointing towards the inside watery environment and the hydrophobic tails pointing to the inside of the membrane. So what we have to remember about membranes is that in this way, they can form, you know, if we're looking in 3 dimensions, they can form this kind of sphere, spherical shape, all right, spherical shape, and in this way they can form a barrier between the inside contents of the cell and the outside contents of the cell. And that is really quite advantageous because then the cell can maintain the conditions that it needs inside, provide, you know, concentrating all the things it needs for essential biological processes like respiration, protein synthesis, DNA replication, transcription, translation. It can keep all those things that it needs inside in this environment, okay? And it can also control the things that go out. But as it is, this membrane can't perform a lot of functions. It's not really well adapted to do that role. All it is a container at the moment, all right? So it needs to do many other things like regulating things that go in and out. It might even need to be able to sense its environment. And if it wants to do those things, it needs to have more components in it. And that's really going to form the next part of our discussion, okay? On its own, this membrane is too permeable. Water can go through it by osmosis, yes it can. However, apart from that, not a lot of other useful things like amino acids, glucose, other things like that can't get through it. So how do living cells achieve all that? By having additional components, and that's what we'll be talking about, what they are, what they do. Okay, so remember, this is called the fluid mosaic model, and essentially it has this layer of lipids. They can move around, you know, kind of towards you and backwards to that side, but not up or down that much. And in this layer, they can also move, so they're kind of moving around all the time, but only in these kind of 2 dimensions, okay? Towards you, to that side, backwards, but really staying in that plane, okay?
sorry. Stop.